Hello everyone, I am most pleased to be interviewing Dr. Emani today, otherwise known as Chandra, who is a research scientist at MaxGene Bioscience, and he specializes in crop transformation. So Dr. Emani has been involved in various genetic engineering projects with various crops, and now is involved in researching the genetic modification of cannabis plants. With that, I would love to turn the time over to you, Chandra. Can you tell the students more about your background?
Sure. So I started off my career as a plant scientist way back in 1992. That's when I started off as a grad student. And I was in India at that time. Molecular biology, especially plant molecular biology, was still at its infancy, I would say. So I was one of those few lucky ones who actually not only got into the research, but also built the lab as we speak. So we used to have a fancy time getting all these packages from across the United States or Europe. So we used to unpack these fancy instruments like electron microscope or PCR machines and stuff like that. So I basically set up one of the first plant molecular biology research labs in India. So that was the most thrilling part. And I started off working with peanuts. And at that time, Rockefeller Foundation had a big presence in India. So I was a junior research fellow of the Rockefeller Foundation. And my project of interest was with peanuts, where I built gene libraries of peanuts. So to put it simply, what we do is we grow peanut plants, isolate the DNA, and then we cut it using restriction enzymes and put it in a vector and make the whole genomic library and seed in a library. And then that library can be used to fish out our genes of choice. So some of the genes I worked on was fatty acid desaturase genes, because peanut oil is a very good oil to cook, but it codes for high cholesterol when you consume it. So we used to take out those genes and then knock them off. The CRISPR wasn't around there. At that time, what we used to use was what was called RNA silencing. So we used to take a copy of the gene, engineer it in the reverse, put it back in the plant with the hope that like cuts like kind of approach. So peanut was one of the first crops I worked with. And then in 1997, once I got my PhD, that was the time I came to the United States, again on Rockefeller Foundation fellowship. I came to Texas A&M University, and there I worked with, you name it, like I worked with cotton, tobacco, soybean, rice, and then wheat, sugarcane. And then from 97 to 2009, that was the time I was at Texas A&M. And that was when I became a full-fledged plant genetic engineer. So lots of genes put into plants, like there were genes which used to make cotton plants fungal resistant. A place in India where I did my undergrad. And this was the place where I did my master's. You see, I worked with amaranth at that time. But the more interesting thing was in PhD, but let me kind of come down to show you how genetic engineering can actually be a dream come true. This is cotton plants, which I transformed with GFP, which is a green fluorescent gene in jellyfish. So when I introduced that into cotton, you can see how the leaves and the fibers of the cotton, they get expressed with GFP. And then most important thing is, I said, fungal resistance. So you can see on top of this slide, you have cotton leaves on which I put the fungal cotton plugs, and you can see all the leaves completely turning. This is called leaf rust. This is what happens to cotton in the field. But you see below these leaves, which are still green, though I put cotton plugs on them. This is because I sent in a gene called chitinase. Now, every fungus has this outer cell wall called chitin, and chitinase breaks that. So these plants are naturally resistant to fungus. So this was some of the dream come true for us, to have environmental friendly fungal resistant plants in the field. We don't need to spray pesticides in the plants. So that's what genetic engineering does. So after this, in 2010, I came to Western Kentucky University. There I started working with herbs. This is like basil, mint, and ginger. And this was because these plants also have a lot of compounds which are anti-cancerous. So I used to extract out those compounds, put them on cancer cells, and I could see that they just stop tumor growth. And these are like, they don't have any side effects. Like when you do chemotherapy, you lose a lot of hair, right? But if you use these plant-based compounds, they specifically target cancer cells. So I got interested in that aspect as well. So 2010 to 2023, I was at Western Kentucky University. So I was a full-fledged academician. But then I loved teaching, so I was there. But then I started looking at my lab work. I was missing out on lab work because this university was mostly a teaching-based university. My research was taking a step back. So that's when I thought maybe I should switch gears to see what's out there apart from university life. So that's how I ended up applying in the corporate industrial world. And I came to this company called MyFloraDNA, which is located in Sacramento, has a subsidiary called MaxGene Bioscience, where we use CRISPR, which is a technique which we use to edit out genes as opposed to genetic modification. So that's how I came to work with, and the principal crop in MaxGene and MyFlora is cannabis. So that's where I am. MyFloraDNA was a company which started off in 2021. And what we do here is, if to put it in a simple language, our founders saw the momentum that is going towards cannabis, right? Let me kind of jump ship here and show you what cannabis does. Hemp and cannabis, a lot of people don't know that marijuana is the 6th most cultivated cash crop in the United States. Oh. So just look at that graph over there. You have corn, soybeans, hay, wheat, cotton, and then comes marijuana. And it's what you're seeing, the figures there are the billions of US dollars that are in the market. So marijuana commands, as of 2022, about 5 billion US dollars every year. And I'm talking about just cannabis here. No, I'm not even talking about hemp. If you combine that, it might jump ship, maybe the 3rd most crop. So why is this a crop? First of all, it all started with hemp. Hemp has a crop value. It's photosynthetically efficient. It actually is a crop. If you plant on the field, forget about its products. Hemp seeds are very nutritious. Hemp seeds, if you consume, even if you go to Walmart now, if you go to the gluten-free section, you will see that there are hemp seeds. Why hemp seeds? Just 3 tablespoons of hemp. 3 tablespoons of hemp every day will give you your entire protein serving for that day. That's how nutritious that crop is. So you have people sprinkling hemp seeds in salads when you make those green salads, and you can just sprinkle and eat them. And hemp seeds are very tasty. They have like an almond nutty flavor. But more than that, hemp fiber is commonly very important in terms of it's much better than jute. So you could be using that in a construction material. Then hemp leaves, they're so photosynthetically efficient that they fix carbon dioxide and they capture carbon much more efficiently than any plant out there. So it's a climate friendly crop. So the fiber, grain, and the phytochemicals, this is what makes this crop out there. We have in the East, we use hemp fiber to even manufacture paper and clothes. So it might someday rival cotton in terms of it being much better in terms of the resilience and recycling abilities, right? And also that's the crop value. When you look at the traits, it's a day neutral crop. The flowering time is so efficient. No cross-pollination involved. So if you have some variety, you could be using that. Fiber, actually hemp oil. Hemp oil is a very healthy oil. And it's tall, straight, bushy, so it can be a forage crop out there. It's naturally disease resistant to many pests. And the secondary metabolites are much better in terms of their medicinal value. Now let's come to cannabis. As I told you, 6th largest crop. The phytochemicals in cannabis, they are the potent painkillers, inflammation treating chemicals. The secondary metabolites, when you look at them, their medicinal value is up there. We have evidence now that it can treat epilepsy, heart disease, maybe cancer, because everything starts with inflammation. The problem is it's a scheduled crop. So what makes it that scheduled crop is the THC content as we know it, right? But if we can bring down those levels naturally, right? Breeders have been trying to do it, but then they have to, when you do breeding, it's like, it's not just targeted genes which we target. So out there now is our interest in terms of editing out those genes step by step, and there are traits we can target, like THC is the most obvious, bring down the drug content, and then it can become a Schedule 3 or Schedule 2 crop or whatever. And then disease resistance, a lot of viral diseases. We have drought resistant traits to take care of. Then the color, the yield. There are certain things like some of the people would like the skunk-like smell to come down. Again, it's all metabolic pathways which we need to target, right? So you can see just the $1 billion in terms of the states, the various states. California tops, and that's one reason why our company is located in California. So what do we do at MyFloraDNA is we started off as a genomics company. So from around the nation, we get leaf samples of cannabis. And what we do is we do a detection process based on molecular diagnostics to show that viruses, if they are present, right? And we tell farmers, okay, just based on one leaf you sent us, we can tell you what viruses potentially are present, and we even suggest them as to what to do with that. The other very important thing we do is cannabis has been a folk medicine kind of crop with no genetic stocks available. Like there are no identity, a molecular identity. Like if you go to corn or soybean, once there is a crop, we know a company or a farmer who owns that. Cannabis is not like that. It's a free-flowing crop. So when we do the viral detection, we also give it a molecular barcode. It's almost like the person who sends us will get back what we call as an MF detect barcode. So now he owns that stock. Not that he would patent it, but now we have a proper genetic base for every cultivar out there. So we are doing that. We expanded out, like we do like 300, already we crossed around 300,000 diagnostic samples, 200,000 fingerprinting samples. That's when we realized that could we be using this profits which we are generating with genomics into managing a CRISPR editing platform in cannabis, right? Can we do gene editing in cannabis? So that's what gave rise to MaxGene Biosciences, which is a dedicated facility for tissue culture and transformation, right? So CRISPR actually is a very, again, let me kind of, when you look at CRISPR editing in crops, it's a commercial reality now. Like this pretty much shows you what USDA has approved in terms of, as you can see, all these crops, alfalfa, banana, strawberry, soybean seems to be the biggest crop, and then pennycress, then you have blackberry. Things are being targeted, right? Let me kind of tell you what CRISPR is all about, right? CRISPR stands for clustered regularly interspaced palindromic repeats. That's what it stands for. Let me break it down in a very layman's language as to what this all is. As the name says, what happens is this was discovered in bacteria when a virus attacks a bacteria, right? So look at this finger of mine. This is your bacterial DNA. When the virus attacks it, right? What the bacteria does is it catches the virus sequence, and it's almost like it gets a mugshot of a criminal, right? And it stores it. How does it store it? It clips off a sequence from the virus and makes it part of its own DNA, right? And that way, spread over the bacterial genome, you have these small clusters. So that's what they say, cluster, regularly interspaced, because they are put in regular interspaces in the bacteria, right? And then the palindromic, the name palindrome is, if you read the DNA sequence, they read both same way from front to end and back to end. And then there are repeats. So why does the bacteria do it? It stores these, so the next time the virus comes in, right, it uses what is called a guide RNA and captures the virus. And then as soon as it captures, as you can see this blue thing here, there's a protein called Cas9. So it's CRISPR and Cas9, and that particular target chops off the DNA. So the second time the virus comes in, it chops it off based on the mugshot it took earlier. That's CRISPR, put in a nutshell. Now, what do we do now? This shows us that any target DNA can be targeted specifically. So now we took that particular guide RNA DNA motif and we replace the target DNA with whatever target we want. Say, for example, in your DNA, you have a certain DNA sequence which needs to be edited out. It's almost like you want to do a spell check. If you compare your DNA as a long sentence in a book and you have this spelling mistake, you need to get it out, right? Instead of going through the entire thing, it's almost like when you do Microsoft Word, you have an auto spell checker correcting your DNA. That's what it does. So this guide RNA is the most important thing. We can take this guide RNA and take this target, and this will go to a DNA sequence within any entity. It can be an animal DNA, plant DNA. Once it goes there and detects that, Cas9 will take over and chop it off. So this, in a nutshell, is how we can specifically target a sequence within a plant. DNA within the plant, which makes the plant do something which we don't want, right? We chop it off, and then the DNA repair system will fill up the space, and the plant is now having a new character. We didn't, here I want to kind of space, a lot of times they say, okay, so is this genetic modification? Is this a GMO? No, sir. Because GMO means we are introducing a new character into the plant. Whereas here, we are indigenously targeting a DNA sequence within the plant, chop it off, and we are modifying the plant. That's it. So instead of introducing another sequence, we are simply editing out things which we don't want in a plant. So that's how it becomes a different character than the traditional GMO as we want it, right? So now, what is it that we want to do in cannabis, right? How do we go about this? So this is, in essence, the R&D process which we do, right? So we have an ideation. First, we brainstorm. What is the target we want to modify in a plant? So the first thing is, once we do that, we go after what is called scoping and Cas licensing. Because everything we can do in a lab, we can construct the DNA, but the Cas9 is owned by certain agencies, certain companies. So we contact them and say, okay, we are now ready to do Cas9 editing. Can we have that license, please? Once we do that, we go about designing a protocol. How do we get into the plant? I just said we just introduce something into the plant to edit it. So we have to go through that protocol. I'll come to that in a little bit. After that, stage 3 is we edit. Stage 4 is we get a plant with the edited character. So all this takes around 6 months of your time. And finally, it goes into the field where USDA and EPA steps in, and then they approve this target, right? What is it that we can do with this whole process? How do we go about it? Here comes a very important aspect of this whole process, which is the tissue culture and transformation. Again, let me tell you a little bit about tissue culture. It all started with this great man. His name is Gottlieb Haberlandt, as you can see, 1854 to 1945. Gottlieb Haberlandt told us that to take any part of the plant, and you can get a whole plant out of it. Like you take a leaf, put it in a hormone infested plant media, and from the leaf you can get a whole plant. You take a embryo, you can get a whole plant. So that's what is called tissue culture, because this is what is called the concept of totipotency. As the name says, toti, total, potency, the potential. Every part of the plant has the potential to become a whole plant, right? So you don't have to wait for the seed and then, but this is the tissue culture part. Now, Haberland, fun fact, he shares a very great legacy. His father, Friedrich, was the person who actually introduced soybean to the world as a crop, right? And his son, Ludwig, actually invented the birth control pill. So this entire family is one of the fantastic scientific families which nobody knows.
Yeah.
But yeah, so this is like a fantastic thing for the students to appreciate as to where we get all these geniuses from, right? The second set of geniuses I want to introduce to you are Toshio Murashige from Japan and Fox Skoog from Germany. They both came together in the United States to actually formulate a media, a tissue culture media, where you put any part of the plant and you get a whole plant. So we have what is called the MS medium, which we use routinely. M stands for Murashige, S stands for Skoog. So I always have this episodic historical thing. I want my students to see the real heroes, like who actually made the tissue culture all exciting. So what we do then, we then go to the plant and we choose what is called as an explant. Now, in this slide, you see A is how the cannabis or hemp seeds look like. We germinate them, we get the shoot coming out, right? That's the first thing called the hypocotyl. We can cut that, get a whole plant from it. So we test various explants. Which one is the best one which responds to a tissue culture? We go after a hypocotyl, or we can go after a shoot on the cotyledons. We can go after the first leaves, right? Whatever is put on a media which responds and gives us a good plant. So in our experience, where I looked at literature, they said cannabis is very difficult to transform. It's a recalcitrant crop. I said, I worked with worse crops. I mean, I worked with crops like sorghum and rice, which were more recalcitrant than cannabis. So to my pleasant surprise, within 2 months of my coming to the company, I found that I could actually get a beautiful tissue culture response with all these explants. And then what I saw was the most potent one was the cotyledon. As you can see, that's the cotyledon, how it looks like when I take it out. And when I put it on the media, you see those small primordial plants coming out of it, and then a whole plant. Now at this stage, at this cotyledon stage, what we do is we put in what is called as an agrobacterium. An agrobacterium is a bacterium which can get into the plant and it can clip out its own DNA and put it in the plant. Right? This is what is called a transfer DNA. So this agrobacterium is a natural genetic engineer. It's not a bacterium which gives disease to the plant, but it gets into the plant because it is selfish. It wants to make certain amino acids called opines. So what do? We go into the agrobacterium, we let the things of the agrobacterium which makes opines be, but the rest of the DNA is for us to modify. We put in a character which we want to introduce into the plant. So we take the agrobacterium, put it on the cotyledon, it gets in, introduces our character into the plant. That part is what contains the guide RNA and the targeted DNA sequence. Why didn't somebody do it before? It almost seems like the more experience you have with various plants, you try out various hormonal combinations. And then we narrow down to a particular relative of the plant, which is more closer, which in this case was hops, and then hemp, and then cannabis. All these come under cannabis. So as you can see, we germinate seeds like this. We dip those in agrobacterium, like it almost looks like this, right? We can sometimes clip out the hypocotyls separately. So both of them, on the left-hand side, you have the cotyledons, on the right-hand side, you have hypocotyls, right? And then the agrobacterium gets in, and when the plant regenerates, along with the DNA which we keep an antibiotic resistance gene, right? This is something which we use to select our transgenic plants. So in the media, we can put an antibiotic, right? And the plants which grow are the only ones where the DNA transfer has taken place. So that way, we will select only the plants which have the gene inside them. So this is called a selection media, right? Sometimes we put in what are called reporter genes. This is a gene called DS red, right? So you can see the cotyledon now glowing with DS red. So it's a visual marker. So before we get in to our actual gene of interest, we test it with what are called reporter genes to see if the system is working. So as you can see, DS red expresses. And then we go to the whole process. So we go through the explant. We go through what is called co-cultivation, which is nothing but we take all those seeds, dip them in agrobacterium for a certain time, right? We allow the agrobacterium to infuse into the plant, right? And then we select whatever tissue we want. Sometimes we select what is called callus, which is dividing cells from which the plants can regenerate. Then we have plant regeneration. So as you can see, it's a process which takes around 55 to 65 days, right? But at the end of the 65th day, if there is a client who had given us a plant, we can ship him with an edited plant. So what are some things which we did? Proof of concept studies, right? These are some things which are not commercially important, but can we show something that we edited, right? You see on the left-hand side, you have a regular plant regenerating, all healthy and green. But look at the plant in the middle and the right side. We have actually edited out what is called a phytoene desaturase gene, which actually codes for chlorophyll. So as you can see, all the leaves are bleached out. So that shows us precisely we targeted when the rest of the plant is growing. That means the rest of the plant is fine. Only thing is it is now edited out of the chlorophyll, right? Another thing I can show you is stomogen. Stomogen is a gene which reduces the stomata. And as you can see, again, the control plant looks all good, but the left-hand side, you see the leaves are kind of shriveled out. Stomata count has gone down. Right? This is something we can target. And how do we test them out? We kind of do the PCR of the plants itself, which is like, is the gene present or not present? So when I tested out all the transgenics, the brightest bands are the controls, which is the plasmid which I put in. But you see these faint bands in the plant. That means the gene has gone into the plant and we can amplify. So this is how we test it. And then we do what is called a sequencing to see. So you see that's the top thing is the reference DNA. And I compare it with my transgenic plant. You see the top stretch. There's a whole chunk missing here. So that has been edited out. So the sequence will tell you that, okay, this is the gene which we targeted. And you see all those dots there. That sequence is gone. So this is how we can test the plant out, right? So that in a nutshell is what we do and what we want to target. I haven't, but Ajit Anand, who is our chief scientific officer, he's come with a wealth of experience from other biotech companies. Okay. He is very cognizant of the fact that there is a lot to do at that stage, right? But when you look at the amount of crops that are coming out right now, like recently we saw somebody has CRISPR edited the mustard greens, which don't taste so bitter anymore. You know, they actually, and then there is a mushroom crop where the CRISPR editing has done for lessening the browning. All those have been approved.
The PPO, right? PPO mushroom.
So, and a lot of traits they are targeting, like disease resistance, for example.
Yeah.
The amount of time that used to take classical breeders used to be measured in decades.
Yeah.
GMO brought it down to a couple of years or more, but then GMO had all this backlash, which we have to worry with. So now we call this NGT. It's like new genomic technology as opposed to GMO. This cuts down the time a lot because we are not modifying anything in the plant as in getting a foreign gene into the plant, but modifying just the indigenous inside. So cuts down. So the regulation time also in terms of the field trials and the general acceptance might be much faster. CRISPR not only edits out, but it can be also used a tool to insert whatever characters you want. So that is something which is possible.
Okay.
Because ultimately what happens is once you introduce or edit out a character, the non-homologous DNA repair mechanism of the plant takes over and it stitches whatever DNA you put in or whatever gaps you left. So in either case, you get to a stage where you can not only edit out a character, but introduce a character. Again, we can't have like, okay, give me something and I'll give it to you like in 6 weeks kind of a deal, right? It has to go through a lot of research in terms of what exactly are you doing to the plant. So as one of my mentors, the great Nobel laureate Norman Borlaug used to say, he was the father of green revolution. Everybody is like, why is GMO not being successful? Where are the products? And that guy, once he got so irritated, and he said, you want to have a baby without the labor pains. You have to wait. You have to undergo that whole process. It's not like I give me a character, put it in, stitch it, and I'll get it out in the market. So you have to be patient for that. But once it comes out, it's like the next best thing after fire and sliced bread or whatever. It's like you can't live without it. So, but you need to be patient. You need to be really looking at what implications you have in terms of, especially when you're looking at things which we consume. We can't just hasten up and say, okay, I'm going to give you a product tomorrow. So, but that's what I say. And it won't take time. It won't take the time as in we have to wait for decades, but at least some years, maybe 5 to 6 years to see how the process works out. Proof of concept studies, when they are done in especially a great system like yeast, that means that they are going to work in a eukaryotic system, right? So the next step is, but again, cannabis research is so regulated that the university can only offer proof of concept. And we have to go for, like, just I'll tell you how rigorous this whole process is. When I got into this job itself, right? When I'm entering MyFloraDNA or MaxGene, we undergo screening by both FBI and CIA. I was like, wow, makes me feel so important. But that's how much the regulation is. And then once we do these things in the company, we're not allowed to take these plants out into the soil. We can only do tissue culture edits. And then there is a separate license we have to acquire to have a greenhouse where we actually take those. So there, even the length of the plant is regulated. Like, okay, if they say, okay, you can have only 6-inch plants in your facility, that's it. Even 1 more centimeter above that, it's a problem. But then we partner with people who already have a cannabis license made to grow them, because definitely we want to see our plants grow and get to the next stage, right? So all this is very rigorously like regulated, and for a good reason, because we don't want everything to be out there, especially with a regulated crop. Even for the general crop, for example, like sorghum, for example, there is a danger that sorghum, once it's modified, say we are modifying herbicide resistance. Sorghum naturally cross-pollinates with this weed called Johnson grass. If this pollen jumps into Johnson grass, we are creating super weeds in the atmosphere. So do we really know what happens once the plant is out there in the wild, right? So that is something we need to be really cognizant about. So all this needs to be taken care of. Right? Yeah.
Well, I have to say that's one of the few valid arguments that I've found against GMOs is that, if we introduce it into the wild, then it can, cross-pollinate, and then we'll have plants with genetics that we didn't intend initially. And so I do think that is a valid concern. So it's, it's good
but then that is something which can be easily solved. What we do is, whenever we have GMO crops, what we do is we have a crop with GMOs, we are field with GMOs, and then we have what are called buffer crops around, which are like naturally cross-pollinating crops. So we kind of balance out, even if there's cross-pollination, that is the crops which will take it. Then we take them off. Solvable problems. But what happens is the anti-GMO crowd jumps in on the negativities without looking at the solutions. And again, when we get it's like almost saying, okay, we have so many road accidents, so just ban all the automobiles. Let's walk to work. Not going to be practical with the growing needs. And the need of GMOs and now NGTs is, it's very grim when you look at the amount of cultivable land that's going down. Right? Next 50 years, listen to this. we have to produce as much food as what we have thus far. Agricultural revolution became 10,000 years ago. The amount of food which we produced and all that needs to be close to what we'll be producing in the next 50 years. Where will we get all those arable land if we have to go through traditional agriculture? So we do need things like tissue culture and transformation to just meet the demands of the growing population and the food we need to produce. So we can't just say, okay, this is bad, so ban it.
Right.
It's like, yeah, I can walk to Europe if I want to, right? But I don't have that much time, right? So why do we have Boeing or ships, and why do we invent the wheel or whatever? But when you are in that period of time, it's very difficult to convince the crowd. Maybe when we were in that time around, automobiles were seen very dangerous, but now we can't live without. So it's the same thing to any new technology. We need to kind of go through those things. So yeah,
it just takes time for people to acclimate and understand what it is, I think. And yeah, but we're definitely not really in a position to get rid of tools right now in our tool belt when we've got so many people and limited space and resources. So yeah, I see where you're coming from. Absolutely.
You have to choose between what you want to do and where your excitement lies, right? So first things first, when you want to get into a career like especially plant sciences as a researcher, you need to equip yourself a lot, right? It's not just, okay, I got my master's, I got my PhD even, and then I look for a job. No, you need to be patient, maybe a couple of years after that. Of course, I did a very long postdoctoral career, but at least once you finish out your grad school, invest at least 2 or 3 years as a postdoc somewhere. Don't jump in. If you want to be perennially successful and you want to sustain that excitement, right? Like one of my personal heroes, James Watson, the discoverer of DNA, said, you need to be significantly underemployed before you do something significant. So don't be, right after your PhD, if you don't get that big bucks into your bank account, don't be discouraged, because yes, you need to be a poor grad student who lives on ramen noodles in a small apartment for some time. But why? Because you're equipping yourself with a lot of, so look for those labs or companies where you can get those, right? Now, there are two options for you. One is academics. Academics is a very good thing in one way, like you get through what is called a 5-year probation period as an assistant professor, and then you get what is called tenure. Tenure is like you're there. Once you become an associate professor, you're tenured, you won't lose your job, you're very stable. But that's a big misnomer. Tenure is stable, yes, but then tenure is also a place where your excitement is dampened. You do the same thing over and over again, and pretty much you feel. That's what exactly I found myself. Yeah. Academic jobs are not that great economically. If you're a bachelor, perennial bachelor all your life, great. But if you have a family and then you have kids coming in, maybe you want to think about creating a right balance. Like maybe you get a wife who's also an academician. So if both of you are professors, great. But if you're the sole breadwinner, then corporate world is the best place to be in terms of the dividends, the economic dividends. When you're in a company, you get stock options and whatnot. It's a much better pay. But you have to be prepared for this. Corporate jobs are at will. Like I can get a phone call right now as I'm going to this Zoom thing from my boss and say, hey, we decided to close the company. Oh, it's not as bad as that, but I'm telling you, it might be that, because corporate world is done with profits. But how do you do this? How do you come over this? You need to be building your networks. Like as soon as you enter the corporate world, start contacting people, so that a stage may come when you have to shift companies, then you have this wherewithal to tap into your network. So that's the balance you're looking at, like a stable, cushy job where you want to be in, academics, an excitement driven, cutting edge place where you are excited to get up and go to work, corporate world. But in both cases, right after your master's in your graduate school or PhD, invest at least a couple of years, maybe 2 to 3 years in a good postdoctoral position where you learn the techniques. And then you need to market yourself, saying that I am this expert. You don't let your resume read, but you need to get out there and tell what you want. And then once you do that, you are in a constant phase of reinventing yourself. And then again, sometimes they say mammalian field is much better than plant field. Yes, because plant science is so much narrower. But then is the excitement less? I would say as a plant scientist, you're more excited because mammalian world is completely saturated. You have so many competitors. Plant science, though the opportunities are less, the amount of contributions you can make to the field is much higher. So have that balance. So yeah, I would very much want you to look at the success stories of who the real inventors were. For example, CRISPR itself, Jennifer Doudna. There is a beautiful book out there. It's called The Code Breaker,
Okay.
written by Walter Isaacson, right? Buy that book.
Okay.
There is
I'll have
another
to do
book
that
called
too. I haven't read it, so I need to do that.
Code Breaker, required reading. It's a monumental book. It's not just about Jennifer Doudna. It's about a little girl who read a book called Double Helix by James Watson, right? Because her father thought it was a scientific thriller or something, because it reads like that. So you would know what to read too. Double Helix is a beautiful book. It's like a 120-page book, which will really get you excited into the field, right? And it also tells her rivals, the rival research teams. It almost reads like a thriller. I couldn't put it down. It's a bulky book, but when you read it, right? The other thing is, always when you look at certain discoveries, try to go Google as to who the discoverers were, and it will make you blow your mind as to how those things people actually came to those discoveries. This historical aspect is seriously missing in the classrooms. We just read through a lot of discoveries without actually knowing the excitement behind it. Sometimes those are so inspirational, right? Citizen scientists, because these things, without knowing the historical aspects, the regulatory aspects, technological, of course, they get in the classroom. If a man on the street asks you, what is it you're doing? You should be able to tell in a language you would understand. And you would do that only through reading these kind of articles. So that's one thing I emphasize on the students. You may not be a biologist, but you need to be a citizen scientist. And there are so many people. So if you read Code Breaker, you will understand how that the guy who wrote it was a journalist in Time, Walter Isaacson. But the way he dwindled down to get the science to even your grandmother can understand. That's the best part of science. So read that book. Time you want me to get back to the class with any questions the students might have, like after watching this, I'll be more than open to ask all the students to feel free to just email me. See if they want to ask more, I'll be glad to. I'll never be, even if I get a million emails from students, I'm so happy about it because I want to be in that touch with them. And yeah, any opportunities you want to discuss about, not just within the corporate world, if you want a beacon to be shown towards the academic world as to how to go about it, I'll be more than glad. So yeah, you won't be inundating or irritating me with your emails. Even if you may email me, yeah, just give me time to respond to them. But every student out there is welcome to be in touch with me. So yeah.
Well, thank you so much for that. That's very generous. And thank you again for your time. I really appreciated this interview. So
yeah, thank you so much, Sarah, because once I got here, sometimes there are times when I miss classroom. So any opportunity to get back to the classroom.
There you go. Yeah, well, we're happy to have you back in the classroom, and this will be excellent for the students. So thank you again.
Okay, thank you so much. Yeah.